Hello everyone, good evening and a warm welcome to this webinar. We are MedGenome, the leaders in genetic testing in India. My name is Joseph Victor, your host. This webinar is part of a series of webinars on the essentials of genetics in the 21st century. Before we start, I wanted to thank Dr. Narayan Saha for providing us this opportunity to present to this audience. And we look forward to a very engaging webinar. And I also wanted to inform the audience, if you have any questions or comments, you can type it out in the Q&A window of the Zoom webinar interface, and we will address them at the end of the session. And we will also be sending across a recording of this webinar, and in case you miss out any points or want to revisit any points.
The main part which I wanted to discuss today is probably the next generation sequencing. So we can just breathe deeply and then I can start the next part so that the previous thing is little absorbed and then we go to the deeper things. Okay. So I will just start with this history of the Human Genome Project. It began in October 9. You can go to this website. It has recently been revamped. They have a lot of information on this. And it can just be that it started in 1990 and completed in 2003. So almost 13 years it took to decipher the human genome, right? To confirm that how many billion base pairs we have. Initially, it was just an approximation that maybe we have this many base pairs. Maybe we have 30,000 protein coding genes. We were not sure. But at the end of 2001, when the first draft came out, it was so exciting to know that, yes, we are now knowing that humans, they are how identical yet different, and then how close we are to the other chimpanzee genome or the orangutan, and yet we are different. So just a 1%, or if you can see humans, just 0.4% difference, and yet when you check with 3 billion base pairs, you know that we have a lot of variation, which can explain our differences in the eye color, hair color, sizes, shapes, eye shape. So these inter-individual changes make us what we are. But you should visit this site. It is awesome how they've explained everything, how it started, who were the founders, and how they went forward after that. So there is an pre-HGP area and there is a post-HGP area. Again, like PCR changed biology, probably Human Genome Project changed genetics. So right from 2001 to today, if we see, you can see that there is a lot of changes in science, in chemistry, in optics, in fluidics, in bioinformatics, and it has brought down the cost of doing the genome testing from as high as 11 years to less than 24 hours. Probably it is time now to get the human genome in $1,000, which was the dream of the people who started the Human Genome Project, probably that we want to give the genome at $1,000 only. And it is going to be realized. I can feel it, and I'm really excited to inform you, yes, this is becoming a reality now. So it was a serious paradigm shift, like something taking 2,000 machines and equal number of people working on those machines and 13 years to get 1 GB raw data, right? And spending almost $1 to $2 million to get this data, compared to today's time when we in 4 to 5 hours and spending around $50 to get 1 GB raw data from one machine by one person. The same amount of data. You can imagine the paradigm shift. So first generation, now we are talking of next generation sequencing, but we have to know what is the first generation sequencing. Sequencing is basically knowing the DNA sequence. So when you have a telephone, you have a smartphone, and you have the next generation phones, and you have the next generation. So it is just the change in technologies. The purpose or the principle is probably the same. So initially the first generation sequencing was by Maxham and Gilbert method. And then what is gold standard, or which is being used very commonly, is the dideoxy method, which was discovered by Frederick Sanger, and it is still being used as the gold standard. So again, in Sanger, you have to know what you want to test. So that is probably the limitation of Sanger, that you want, because you have to prepare primers for that area, for that region, so that you can cut that region, amplify that PCR product, and then you use primers which will give you the sequence by capillary electrophoresis. Now this method is also changed. First it was by radiology, then capillary electrophoresis, fluorescence. Different techniques have changed even in Sanger methodology, making it more easier and more user friendly, more interpretable. And we all know what a chromatogram looks like, right? So these are different color fluorescent dyes which will give you the peaks of ATGC. So green, blue, red, or purple color is being used to know the different ATGC nucleotides, and it is a single run. Okay, so you are just doing a single run to check for the presence or absence of variant compared to a reference control which is run along with it. So same temperature, same annealing temperature for the PCR. So the control and the patient is run together in the same run, and then they are compared to see for the peaks. So this is the first generation sequencing, and indications are still there. It is not an absolute technique. It is still the gold standard sequencing method. It is not without errors, but still it is very low error rate. So it is still used for confirming the known or confirmed variant by some other technique, and we can use Sanger to confirm it in other members at a much lower cost. And prenatal diagnosis, we always recommend that it is better to do it by Sanger if it has been, the variant has been detected previously in other family members. And now the use of Sanger is also post NGS. So NGS is a very robust technique, but still when it was being evolved over a period of last maybe 8 to 10 years, there were certain limitations in NGS, and to confirm a false positive or a false negative, Sanger is still being chosen as the method of choice. So when there are many pseudogenes in a gene, like congenital adrenal hyperplasia by CYP21A2, that has multiple pseudogenes. HBB causing thalassemia has multiple pseudogenes. You all know about SMA, right? So in such cases where NGS is not the preferred method, we have to use either Sanger or some other robust technique which can rule out that whether the variant which is there is in a pseudogene or a true gene. Sometimes there is low depth of the variant. Sometimes there are so many quality checks in NGS, right from sample collection to the data which is coming out, lot of bioinformatic checks at multiple levels. So if there is a QC failure, then you can do a NGS. And to confirm whether the variants found in the proband are truly compound heterozygous or a truly homozygous variant, then we definitely need to do the Sanger in the patient as well as the parents. So these are the indications, and just discussing the limitations. As I've emphasized again, that prior knowledge of the genome is needed if you want to do a Sanger sequencing. So you have to know that I'm dealing with this disease has this gene, and in this gene, what area I need to check, right? So for this, you have to know about the genome. The patient with unknown genetic disease, you can't do Sanger. You cannot do Sanger for 20,000 genes. Each nucleotide, or when we design the primers are around 11 to 15 base pair or 20 base pair. So every nucleotide is costing. So you have to know the genome, and if you start designing primers for each and every exon or intron, it will be too much costly. So, and then there are diseases which have multiple genes. So now we know that there is very less single gene disorders. They are more of single disorder involve multiple genes, right? Or one gene which is causing many phenotypes. So to confirm that, we cannot use Sanger. We will have to do, like in cancer, we have 350 genes of panel. In nephrotic syndrome, we have more than 75 genes. In retinitis pigmentosa, we have more than 90 genes. So if you start doing one gene at a time, it will take a lot of money and lot of time. And sometimes where are large genes, like DMD is 79 exons. If you start doing Sanger, it will take a lot of time and money. And the limited size of sequence. So in as I said, the primer size and the size of the amplicon has limitation when you do a Sanger. But in NGS, you now have long reads also available, which removes the limitation. And as I told, it is labor intensive for large sequencing, or when you want to do multiple people tested together, it will not be effective. I would just share one experience in which for Marfan syndrome, this was way back in, I think 2008 or 9, when I had recently joined PGI, when this father was refusing to have a kid because he lost his father due to Marfan syndrome. His maternal aunt was in UK. She was also affected, but she didn't get married because of this, that I might pass it on to my kids. So he said he wants to do the sequencing, and we found one lab in UK who was ready to do the sequencing for Marfan FBN1 single gene, and it had cost him 1.5 lakhs. But he did it. And when we found the variant, we did the prenatal in India itself, and he had come later that the CVS is negative, and then later he kept emailing me the photographs of his daughter who was born without the disorder. So we know that it could be costly and labor intensive, but when it was time, it also had to be done. I'm sorry, just a minute. So this is a very good cartoon which can explain the difference between Sanger and NGS. So the conventional, because it is probably powerless when we have so many increasing number of genes, and various variations also are different, and disorders are also coming up. Newer disorders and complex disorders are being discovered. So Sanger may not be fruitful, and hence there is the advent of NGS, which has totally changed the you can say molecular genetics in a big way. So modern DNA sequencing technology, or what we consider as the next generation sequencing, is a very rapid and a high throughput technique. So you can run multiples, hundreds of samples together. And because of the increasing quality of the data which we are getting as we are improving day by day, data storage capacity is improving, various bioinformatic tools are improving, pipelines are improving. It is decreasing the price of testing. It is decreasing the time. If we remember the time, there was a time when we used to take 2 months to get one report out. Now it has come down to even 2 weeks or 1 week, we can give a report. And obviously the workflow complexity is decreasing, and the error rate is also increasing, and more data about how it can be used for other variations is also coming out. Sorry, I'm sorry.
Yeah.
So there are multiple sequencing platforms. We should not go deep into it. They are different good-looking machines all around. Made by different service providers, which are having different platforms which they make available. So there are not much changes, except that differ in the design and the chemistries. So the main is that they are going to sequence thousands of millions of amplified molecules parallelly. So they don't go one by one, but in parallel, they are going to sequence millions of amplified molecules, and it is evolving. So whether you are using pyrosequencing, or you are using sequencing by ligation, or you are using the HIN generation technique, or you are using reversible dye element, these are different companies giving different methods of sequencing, and obviously the output. So either you can use luminescence, fluorescence, pH change, and depending on that, you will get a sequencing data. And this is the data which we have to analyze based on bioinformatics. So sequencing principles may be different, but the fundamental is that you have to sequence multiple amplicons. So here we don't need to know, we don't need to have a prior information. We are taking the whole genome and we are cutting it into small fragments. Okay. And then every small fragment is then very short DNA sequencing are taken together and they are aligned to a reference sequence, and what we get is the sequence genome. So we have a genome, we cut it, and then we align it together. So we are splitting it and then lumping it, and then we are comparing it to a reference. It looks so easy, right? That we are just cutting it off and mixing it together. But lot of error can happen. There may be many duplicates. There may be many fragments which are lost, many fragments which may not get amplified. So limitations are there, but there are again many modules which can override it. There are many control checks which can happen, and you finally get some sequence which can be compared. So Sanger, you are doing only one amplicon. Here you may have to go for more than 40, 50 amplicons, but the depth may depend on the technique and how well the target region was amplified. So if you see the workflow, you have a PCR sequencer, you are doing a library preparation. Library preparation mainly means that you are making, you are aligning them with adapters, you are adding the fluorescent dyes and everything, and then you are making clusters of the amplicon so that you can sequence it, and then you can get images. So now this colorful image looks very good, but it is totally ununderstandable for clinicians. We can't share this with the clinician, right? So this goes to the bioinformatic people and the genome analysts who will convert it to something readable for a layman. Here the layman is clinician, because we really don't know anything which is happening around at this level or this level, right? So this raw data which is being churned out from the machine is made into a readable document by a good team of bioinformaticians and genome analyst people. So in short, if I have see, there is a wet lab where the library preparation sequencing takes place. So this is one lab team. And then you have the big heroes of next generation sequencing, which is the bioinformatic team, which will do the sequence analysis. They will detect the variants, they will annotate it, they will remove the duplicates, they will do a quality check, and then it is going to the clinical reporting team, which will finalize the variant, and then you will get a clinical report. Okay, so what is the application? Now we have done so much, we have put in the sample, but how it is useful in clinical diagnostic? So if you consider this as the whole genome, then in today's time, what we do is we look at the exome. Now exome is the coding region. And I've explained before only that there may be introns and there may be exons, but we all know that the coding or the exonic regions are more important for a disease phenotype because they are responsible for coding a protein. So anything wrong in the exons will lead to abnormal protein. And 85% of all disease causing mutations are positioned within a coding exon. So if you just test the exons, probably you will get a pickup rate of 70 to 80%. You might still be missing a lot, but it is enough for a clinical diagnostic purpose, right? So if to know, you are just testing 1% of the whole genome when you're sending for exome analysis. And then there is an in-between thing which is known as the clinical exome. So what is the difference? So targeted gene sequencing is that you're using a set of genes which will cause a particular phenotype, and you target only those genes for a better coverage and a greater depth. Okay. And then when you see the whole exome, now 20,000 genes, and you are using their coding regions or the exons and test screening for all. And when you test for the whole genome, you are using the coding as well as the non-coding regions, right? But what is the clinical exome then? So clinical exome is in between the whole exome and the targeted gene sequencing. Here we choose genes which have a confirmed disease phenotype in the OMIM database or HGMD database or anywhere in the literature where there are good genotype phenotype correlated database, we have taken all those genes and they are not more than 7,000. Okay, so approximately 7,000 genes of the 20,000 genes are being tested in clinical exome. But here the pickup rate would be better because we are looking at a confirmed disease phenotype. So we know that we have a gene, we have a variant, and this variant can cause this disease. When you do the whole exome or the whole genome, you may get variants for which we don't have information regarding the disease phenotype. We know about the gene, but we don't know whether that gene will cause a disease or not. And hence, the chances of getting variants of uncertain significance or unknown significance goes higher as you start using the higher level of genetic testing. So the test which you want to send will depend on the amount of information you want. So you may need only single gene. So you have a confirmed case of thalassemia, you can only send for the HBB gene. You are very sure it is congenital adrenal hyperplasia, you can send for the CH gene. So the data is less, you are doing just single gene and you are getting a diagnosis. But when you are sending for osteogenesis imperfecta or a suspected skeletal dysplasia, which you are not confirmed, you have not done x-rays, or it is a fetus and you have just identified short bones or a large skull or a narrow thorax or polydactyly, you are not sure what the diagnosis is because you don't have the baby in front of you to do an x-ray and check whether it is which type of skeletal dysplasia it is, and it is still evolving. So that time you have to choose panels. So you can use a skeletal dysplasia panel, you can use a osteogenesis imperfecta panel, and you can be ensured that all the genes which cause that phenotype will be covered till date. So, you know, if you come back later on, probably a new gene will be there which will not be covered in that. So you have to know whether when was the last time when the panel was upgraded, whether it was upgraded 6 months back or 2 years back. So gene panels might sometimes be outdated. And similarly with clinical exome, they also need to be upgraded because these single gene panels clinical exome are made by lab based. So every lab may have their own experts who will design the panels or how many genes and how much coverage of the gene needs to be taken. And when you do the exome sequencing or whole genome, it is by the service providers. So again, there might be a little change, but probably exome of 20,000 is the same. But coverage and everything would depend on the service provider who is giving you the chip. So amount of information may change based on which panel you are sending and which service provider is there, what is the coverage, everything would be different in different panels. And you say, let's start sending whole genome for every patient. But it doesn't come to that. Means more data doesn't automatically mean that you're going to get more knowledge. It is just that you are needing more space to store the data. You will get more variants which may be unknown. Even after annotation, filtration, you may still have a lot of variants which you cannot give any significance, right? Because we don't have literature related to those genes. And from those million records, you have to like choose 1 or 2 which can cause your disorder. It needs lot of organization, analyzing and interpretation. So doing a higher level test may not always result in a decision or a diagnosis which you want. Okay, so you have to choose the best test for you and your patient also, because if you're doing 1% of the genome, the cost will also be proportionately less compared to when you do a whole genome. So again, going to the bioinformatics pipeline. So there are various phases, as I can explain to you, that first is how they will get the raw reads. Then they will map it to the reference sequence. They will do an alignment. They will remove the duplicates. And then this will be the first output from the bioinformatics pipeline. So many of the clinicians ask for raw data. So this is what is shared to them. Then this goes to the level 2 or the phase 2, where they compare the samples, they have the reads, and then they find that what is the single nucleotide polymorphisms, whether there are any insertion deletions, whether there are small structural variations present. And so they filter it off based on the mutation or variant which is there. So this will be a smaller file compared to what the first raw read would be. And then they can go to phase 3. Now, phase 3 means there should be a lot of external data which can be used as a filter. So they might need information regarding whether we are looking at autosomal dominant variant, autosomal recessive condition, X-linked, mitochondrial. So whenever you are sending a sample for testing, you should ensure that you are giving as much information about the family history, whether there is consanguinity. So if there is consanguinity, we know we have to look for autosomal recessive genes. If their pedigree is more like an autosomal dominant disorder, then we have to focus on autosomal dominant genes. And we have to see the population structure, whether it is coming from a particular ethnicity. So certain genes or variants in those genes are more common in Caucasians. They may not be common in the Asian population. We didn't have an Asian database, but now we also have that. So we have to compare in the Genome Asia database also whether this variant is common or not. And then you have to compare it whether that variant is a known variation. Has it been reported previously? Whether any functional studies have been done. And all these external data which we put it as filters will increase the significance of the genotype. Based on the phenotype, we can then make it more, we can filter it out, and unknown or unwanted variants can be removed from the analysis. So we also have the ACMG guidelines which we use to classify. I will be covering all these things in one slice, but I just want to know what are the various filters which we use. So we use the public database like 1000 Genome or the Exact, or we use our own internal database to see how common that variant is in the database. And then we have to use in other disease database. So this is a normal population database to see the frequency of the variant. And this is disease database. So whether the variants have been reported in diseased or patients before. And then we try to prioritize the variant based on prediction tools. So some of the variants will be very new. We don't have functional studies or previously reported. So whether that variant is going to cause any change in the protein structure, whether it is well conserved in the evolution of species, and how common it is. So there are many algorithms based on which these tools will predict whether the variant can be benign or whether it can be pathogenic. And then we can have the internal controls or validation done for these variants. And most important, again, it becomes whether we have the same variant in the family. So sometimes we often ask, please do it in the parents, or please do it in the affected or unaffected people. So if there is a new variant and 2 people in the family who are affected are having the same variant, then we know, okay, this could be significant. But if the unaffected person also is having the same variant, then it cannot explain. And hence we may have to say, okay, it is a benign variant because it is not causing anything in the unaffected person. So it might be ignored. And what is MAF? I will again be taking it next, but just that it is the allele frequency in the population. So the rarer the variant, the higher the chances that it will be causing some damage. But the more common the disease in the general population, lesser the chances of it being related to the disease phenotype, unless it is a very common disorder or it is very common in the population and there is intermingling or inbreeding or consanguinity. Okay, so I think if we have to start interpreting the report, everybody should read this article, which is the ACMG guidelines, and these are getting amended also. We had changes in 2018 a little bit, and also in 2020 we have got certain changes. Now they are going to more score based data. So they have started giving some score to each and every finding which you have, and higher the score, higher the chance of it being a pathogenic. So what does this ACMG guidelines suggest is any change in a gene is variant. So initially we all used to call that it is either a mutation or a polymorphism. But over a period of time, they have understood that, and that was based only on whether it is common in the population. So any variant which is more than 1% in the population was called as a polymorphism. But that is not true anymore. So any change in the gene is now called a variant. It could be pathogenic or it could be benign. For diagnostic purpose, we classify either as pathogenic, likely pathogenic, or variant of uncertain significance. Benign and likely benign, we don't even report because it is not going to change to any decision will not be taken based on benign and likely benign. So we try to focus on these three classification. So either the disease causing variation can explain the patient's symptoms. Okay, this usually means that there is a good phenotype genotype match, and the variant wise also it is going to cause some change in the protein which has been reported previously. And something in between pathogenic and VUS is likely pathogenic, that we have little bit of evidence, but we are still not sure. But probably it has been reported previously, it is a partial match, but we need some more confirmation to confirm the pathogenicity. And the big chunk, almost like 30 to 40% cases in any genetic lab will be classified as variant of uncertain significance. Okay, so it is not a very uncommon thing because we are still evolving. We still don't know everything about genomics or variants and anything. So there are certain gray areas where we don't know whether the variant is pathogenic. It could still be benign, but whatever literature is available, we are giving you information, but we might need other family member testing, or probably some more investigations can be done, or some biopsies can be done to prove that this is causative. Okay, so functional studies, without functional studies, without doing segregation studies, or without waiting for more literature to come for that variant, this variant cannot be reclassified without more evidence. So this is the gray zone, and this is still the most uncertain result which we give out, and we are really not happy about it. Even the clinician is not happy, and the worst person is the patient, because he feels that he has been cheated, that you are giving something but still not confirming whether it is yes or no. So, you know, but we cannot ignore it also. So just going stepwise, if we say that, okay, after doing an analysis, we have found around 3,000 variants, then we have to see whether it is well covered. So it will filter out some more. And then we have to see how significant, whether it is a missense variant or a nonsense variant. So still you can filter out many more. And then you have to see the public database and the disease database, and you can still filter out more variants. So just by filtering out at various levels, out of 3,000 genes, you will still have 30 variants. And then you can use the ACMG guidelines and you can classify them. As you can see, major chunk is usually VUS. So it's not easy to find a variant when you are sending an NGS, because it takes a lot of efforts to confirm it. Most important being that the phenotype has to be very clear. What we are checking, we have to know. Okay, so I'm just simplifying few words which will be there in every genetic report. So you have to know what is the coverage of the gene. You have to know the depth. You have to know the minor allele frequency. You have to know the in silico prediction software, and you have to know what is an IGV. So just speaking about it, I'll just inform also, or in the next few slides, I'll show you. So coverage is basically how much of the gene is being covered. So some genes are very easy and they may be covered 100 times. 100%, sorry, not times, 100%. But some genes are very complex. They have many repeat regions. They may have many pseudo genes. So the NGS may not be covering certain areas of the genes. So coverage is the percentage of the genome which is being sequenced, even at zero depth. Okay. So for example, NCF1 gene, if you are sending a report for primary immunodeficiency, you can see that the coverage of NCF1 because of pseudo gene is very less. Similarly, SMN1 will not be covered in NGS. The SMN2, which is a pseudo gene for SMN1, is almost 100% matching except at 3 bases. So it cannot be covered by NGS. So if you are sending a sample for SMA, and you will get a reply that SMA is not covered. So non-deletional SMA will be covered, but deletional, which is due to the deletion of exon 7 and 8, will not be covered by NGS. So you have to know the coverage of the genes which you are sending for, and depth. Depth means how many times the base. So you know that there are it's been multiplied multiple times. So every read, how many times it is being read. So just like Sanger is being read one time, you can have these genes in NGS being read 100 times. Okay, so the minimum cutoff is take that, okay, it should at least be read 30 times. So you can say what is 30x, what is 80 to 100x. So these are the various depths which we take as cutoff, that only if it crosses this depth we will be covering those genes. Any gene which is less depth is of a poor quality and will be ignored. Okay. Okay, allele frequency I have already covered. And what is IGV? So just like Sanger will have a chromatogram in which it shows the peaks of each and every base which you have read. So you can see the blue peak is related to T, or there's a green peak, and wherever there is a change, you will know that here it is homozygous, here it is heterozygous, right? In the same way in NGS, because instead of a single read, we are reading it multiple times. So this is the same fraction of the DNA. If you can see here, this is the FANK-L gene, okay? And this is the chromosome position which they have highlighted here. So this region is being magnified here. And if you can see this, this read is like this is a screenshot, so you cannot see 61, but there are 61 times, so the depth is 61. So the read has been made 61 times, and in every read, instead of C, so this is the reference sequence, instead of C, every read is showing a T. So this means that this is a homozygous variant. So if you see the red and blue, these are the forward and the reverse strands. So we all know DNA is having two strands, forward and reverse. So both strands are amplified, both strands are being read, and in every strand you are having a change. Okay, so this is a homozygous. If it is a 50%, like count is 61, but 25 to 30% are showing C and the rest are showing T, then it becomes a heterozygous. So we have a cutoff, ideally like 50-50, but they have a cutoff a little bit here and there, maybe 70%, or you can take it. But if this is not good, see, you can see certain breakages. So if it is not a well covered area, you might have this depth will be low, you will not be confident, and then you will have to do a Sanger for that. Okay, so when you see a final report, the variant description should have all this. What is the covered area of the gene? So this gene is 100% covered. Then you have to have the zygosity. So whether it is a heterozygous should be mentioned. What type of variation, means a missense variant or a nonsense variant, frameshift variant. It is also mentioned in the result interpretation. Which exon of the gene? So this gene is on which chromosome? And what is the depth? Now you can see this was 224 times read. So this increases the confidence that this is a true positive result. And then this is the chromosome, this is the nucleotide change. So from G it is changed to T, and because of that the amino acid aspartic acid has been changed to alanine. Okay, so this all information is given in a report. Then it also gives information whether it is in a domain. Domain is important because every protein has critical domains, that any change in that critical domain is bound to cause a change in the protein structure or size, shape, amount. Okay, so we need to have the information whether the variant is some critical domain, that also needs to be told. And then we have to know whether it has been reported previously in public database. So this is not reported in public database, means it is a very, very, very rare variant. So not reported means the minor allele frequency is 0. Okay, we take the cutoff as 0.5. And in silico prediction. So just as I told, these are just in silico softwares which will predict. So this variant in this gene is being predicted as probably damaging by one software and damaging by 3 other softwares. So we take like minimum of 5 softwares to see the prediction. Probably one other will say it is benign. The third one will give conflicting evidence. So there are more than 50 such predictions, and daily based on new algorithms, new and new are coming up. So whatever the lab is confident, whatever has stayed for a very long time and which has been accredited, we use those in silico predictions. And then reference codon is conserved across species. The meaning of this is that any codon which is very critical for evolution will remain conserved across all species. If it is a recent change, then probably it is not necessary for evolution. So it may not be very significant. So if it is conserved across all species, means it is a significant codon. Okay. So you can see that so much evidence is needed to classify the variant as likely pathogenic. This all is taken by the ACMG guideline. So we put all these points in the ACMG guideline pointers, and then it predicts whether it is a likely pathogenic. But yet the final line will always be that it has to be correlated with clinical symptoms. Okay. So you can get such variants. Everybody is a carrier of some or these other, like almost in 9 disorders, you may be a carrier of a significant variant, but it doesn't mean that you will have that disorder, right? So you have to be sure that whatever variant is being reported is matching your phenotype. Either you go back to literature, you call the patient again, you check the phenotype and reconfirm, yes, it is matching the phenotype. But if it is not matching, then you have to go back to the lab and inform that the variant, in spite of being likely pathogenic, is currently not matching the phenotype of the child. Okay. So these information has to be, this is how you will read a report. So in clinical settings, it is that why NGS important. Not only NGS, any molecular test is important. First of all, to confirm the clinical diagnosis. But when there is genetic heterogeneity, when there is phenotypic heterogeneity, you need to have NGS. For precision medicine, for carrier testing, for prenatal diagnosis, you need to use, and for all these purposes, NGS is being used. So why NGS is better? We all know that yes, common mutations it can check. It can also check for uncommon mutations. But it is also very important that it is now picking up more mutations compared to the genotyping method where you are doing a targeting testing. So it is 5 to 10 times more mutations are being discovered by NGS compared to the old genotyping methods. So I'll just take 2 or 3 cases before we come to the end of this session. So this child is having oculocutaneous albinism. Now it is a very easily diagnosed disorder. You can see the white silvery hair, hypopigmented skin, almost reddish hue to the eyes, but otherwise the child is good. Okay. But we know that it can cause severe photophobia, photosensitivity. The child cannot go out in the sun. He has to wear a sunscreen all the times. He has to remain in a colder environment also. There can be many complications also. And in India, 40% of the cases with oculocutaneous albinism type 1A carry a very common mutation in exon 2. So first and foremost, what was done was, let us check for the common mutation. This is because it is very cheap for the patient, like just in maybe 4 to 5,000 rupees, you will get a diagnosis, and 40% is quite a big number, right? But this came out to be negative. Now, OCA are again like almost 7 more different genes. So if we start doing one at a time, it will take a lot of time and it will take a lot of money. So we directly went ahead for NGS, and see, TYR is the most common gene for OCA. Other genes are less common, but in TYR gene itself, a variant was found which was homozygous. The depth you can see is 131 times. This is the codon change, G2A, and this is what the protein change is. Okay. So now after doing a common, like we test for the common mutation, ultimately because more number of genes are involved, which is known as genetic heterogeneity, we may have to go for the larger panel or probably the clinical exome. Now DMD we all know is one gene, one disorder. The thing is that it is 79 exons, so it is a huge gene. Okay, and we all know the clinical phenotype. We know CPK will be elevated. We can do the NCV or EMG, and then we had a muscle biopsy option to confirm with dystrophin staining whether it is absent, to confirm whether it is DMD or BMD. But probably now muscle biopsy is absolute, because they feel that the child who is already having some problem in ambulation, if you do a biopsy, he may have to, it progresses the disease faster. So instead of a muscle biopsy, we can directly go in for a genetic study. Here there is a very typical familial case in which you can see females are carriers, all males are affected. So, and these are obligate carriers because more than one generation of males are affected. So all these females whose son or sisters son are affected, they become obligate carriers. So they have to be tested for prenatal. So the problem or the what I can say is multiple variations are present. It is not a very easy that okay, now there is only one gene, let us do one test. The problem with DMD is that it can have deletions, it can have duplications, but it can also have point mutations. Okay. So initially, I had explained about multiplex PCR. So before MLPA had come, there was a test which was two tube based test. Because it has 79 exons, almost 23 exons could be tested in 2 test tubes. Only 23 because they were all in the hotspot region. Hotspot is a region in the gene where recurrent mutations or deletions are known. So just by doing multiplex PCR for these 23 common genes in the hotspot, almost 98% of the deletions could be tested. And rest were labeled as non-deletion DMD. And they were stopped at that because we had no other way to identify that if it is not a deletion, then what else it is. So when MLPA came, multiplex PCR again, as I said, being a cheaper test, we used to screen the child for deletions. And if it came negative, we used to send for MLPA because it covers all the 79 exons. So we are not missing deletion in any exon. Okay. So if MLPA is negative, then we have to go for sequencing of the DMD gene, because as you can see, almost 20 to 25% can have point mutations. Okay. So this is what happens when there are different variations which are known in a single gene. Same is for HBB. So HBB also, or hemoglobin B gene, also has deletions, duplications, point mutations, splice site mutation, promoter region mutation. So a single test will not help you. So you may have to go for sequencing plus a deletion duplication testing for HBB also. So now when MLPA is there, we don't go for the screening test for 23 exons. We directly go for MLPA. No use for doing a 2-level testing and spending twice for the patient. And MLPA is also not very costly compared to sequencing. So I will just show you 3 reports. A person who is affected with deletion. So we say hemizygous when it is a male because he has only one X chromosome, and being an X-linked recessive disorder and one of the exons, one of the alleles having, or the X chromosome having the deletion, it is classified as hemizygous deletion, and it has deletion of 4 exons. Okay, and from 45 to 48, you may have hemizygous duplication. So there is only duplication of 1 exon, but it is also pathogenic because it is a reported duplication. And then we have point mutations. So here there is a termination. TER means termination. So because of this addition of a A in this codon, this protein is getting terminated at codon 18. Okay, and this is X-linked hemizygous pathogenic. So you can have multiple type of variations in DMD. But why you need to do it? Now it is being important to know whether the child is having deletion or a point mutation because of availability of precision medicine. So there are almost 34 trials going on in the world just for treatment of DMD. And 2 of them have been successful, and which are beyond the clinical trial, FDA approved and already in treatment for DMD patient. So one is the 51 exon skipping. So I had showed you a figure. So exon 51 skipping is FDA approved, and but you can see the cost is exorbitant. And the only thing it does is that it converts a severe DMD to a milder BMD, which is a Becker's muscular dystrophy. And but it is still considered that it can be useful for 13% of the DMD population. And the other, the other therapy which is now coming up, which is oral, is again another method in which for nonsense mutation. It is useful only for nonsense mutation. So the company has started coming up in a big way to collect these patients who have this nonsense mutation. So once an MLPA comes negative, these are screened for the nonsense mutation. So almost 10% of DMD patients will have a nonsense mutation. So the same concept is being working for other. So there is justification that if 51 exon can be skipped, then other exons can also be skipped. So based on that theory, probably other trials are also going on. So for precision medicine, it becomes very important that you have to have a molecular diagnosis. And again, if this is negative, like NGS is negative, MLPA is negative, then probably you have to review the diagnosis. Probably you're not dealing with DMD. It could be a similar muscular dystrophy like LGMD or other sarcoglycopathies. So either you may have to go for muscle biopsy or you go for a bigger panel which test for other muscular dystrophy genes. So these are almost like more than 65 genes which can cause LGMD or other muscular dystrophies, which needs to be tested if DMD gene sequencing and MLPA is negative. But you can see that DMD is also covered. I'm sorry, I will just have the pointer. Okay. So you can see that DMD is also covered in this muscular dystrophy panel. So once the MLPA is negative, you can go ahead and do a muscular dystrophy panel. So along with DMD, you can also screen for the other muscular dystrophy genes in one go. Okay. So here you can see this was one child who was diagnosed clinically as DMD, but he was found to have a homozygous mutation in SGCB gene, which causes limb girdle dystrophy type 2E. And this is another child who was diagnosed with LGMD type 2B, having a likely pathogenic variant in 2 different regions. So he's a compound heterozygous for dysferlin gene. And then now coming to VUS, so we all get upset when we get a variant of uncertain significance in the report. But as I told you that almost 30 to 40% of the reports go out as VUS. So this is one case in which he was diagnosed with congenital cholestatic liver disease. So all the phenotypes related to this disorder which can cause a cholestatic liver disease were checked. So PFIC or alagili or liver metabolic disorders, other inborn errors of metabolism like tyrosinemia, everything was, or galactosemia, everything was checked, but we couldn't find anything. So then we asked the clinician to share some additional finding, and he said, yes, the child also has ichthyosis. Now, just by adding one more word to the analysis, we could pick up a gene which causes ichthyosis and sclerosing cholangitis. So when you don't even suspect a clinical diagnosis like this, NGS will retrogradely help you to confirm the phenotype. So it was a termination homozygous variant, but it causes other clinical features also like alopecia, ichthyosis, and leukocyte vacuoles on biopsy. So if all such information is not provided, or we have not even looked for it, probably we'll be missing diagnosis. So putting this variant in the report directly when the patient doesn't have it, it becomes a little skeptical whether we can classify it as pathogenic. because as soon as you say partial phenotype match, the variant is by default classified as variant of uncertain significance. So the gold standard is that you start giving as much information about the patient, evaluate the patient head to toe, do as much investigations you can do for that patient, which is within his capacity, and then come to a provisional differential diagnosis. And then the NGS will also help classifying the variant better. Sometimes you can go back to the lab and give the additional phenotype, and then a VUS can be reclassified as pathogenic. For example, this child was lost due to glycogen storage disorder, and the parents were sent for carrier screening. The child was not there, so the mother was screened, and we found a good termination variant in a disorder which causes GM1 gangliosidosis. But since GM1 and GST is not a matching phenotype, it was classified as uncertain significance. So the clinician went back to the pathology lab, the biopsy tissue was reevaluated, and then the pathologist said, yes, GM1 also might be having similar features and it might have been confused. So sometimes even the MRI we show to some other neuroradiologist, or we take a second opinion. Sometimes they can pick up clues which might have been missed in the previous. Sometimes we may have to do a MRI again. Now, when this was there and the father was also tested, he also had the same variant. Because when two children are dying of a similar disease, it is very likely that it is an autosomal recessive disorder. And GST dying at so early age, so sometimes we can't make 1 plus 1 is 2. So we have to have more information. The same variant was found in the father also, and so this variant of uncertain significance was then classified as pathogenic, and it was tested in the CVS also. And sometimes we may really not get anything. So this is a child who was having osteomyelitis, liver abscess, and they were suspecting hyper IgM syndrome. But in spite of testing for all the primary immunodeficiency syndrome or all the hyper IgM genes which are known till date, we didn't get anything. So there are situations in which we may not get. So it may go out as a none report. But a none report may not always be a negative report. So there is a difference. Probably there are some new genes which have not been covered, or they have not even been discovered. So there are still situations in which 20 to 30% cases we may not get a clinical diagnosis. So what does a negative or a variant of uncertain significance means? Means that either probably you have to first rule out a non-genetic disorder, then whether we are looking at deletion duplications and we have sent the wrong test itself, or whether there is a lack of clinical details and we need to correlate. So then even in that we may get some positive feedback, and we can either convert a VUS or reclassify a VUS, or even a negative report we can reanalyze and we can get one diagnosis. Probably we have sent the wrong panel, we have sent a shorter panel and the gene is not covered, or promoter regions and deep intronic and trinucleotide. We all know that this cannot be covered by NGS. So any variant in these regions will not be covered, and you might get a negative report or a none report. And then probably technical issue, poor coverage of the regions, like I told before, that the region itself is very complex and we cannot have probes for that region, or there are pseudogenes. So these are many various reasons in which you might get a negative or a VUS report. So the question comes at the end of the session that is molecular diagnosis or a molecular testing a must? So being clinicians, right from the first MBBS, we are taught how to evaluate a patient and come to a diagnosis, right? So sometimes just based on clinical features, sometimes based on various biochemical tests or immunological tests, we can always come to a diagnosis. Like PKD, you just need to do a USG, or you will do a biopsy and you know that it is a FSGS, so your nephrotic syndrome is confirmed. So they may not always be required for a diagnosis, but yes, it is required for precise diagnosis. So which gene of that phenotype is being evaluated, or what is the treatment, as I showed that precision testing is done, and then carrier detection and prenatal diagnosis. So perinatologists and fetal medicine experts know the importance of prenatal diagnosis, and it cannot happen before diagnosis which is molecular based. So proband testing, carrier testing, anything has to be done, but it has to be done before you send a sample for prenatal diagnosis, or you can send it together. But a direct prenatal testing for a suspected previous child with so and so, it becomes very difficult to give a classification for the variant, because we don't know the fetus is affected or not, and so we cannot classify the variant even if we find it, because we are not sure whether it was the same variant in the affected child. So you can do a carrier screening before you send for a prenatal diagnosis. So if you just see a molecular testing algorithm, this I've picked up from a very good journal. I'm sorry I didn't write the reference here, but you can see that when the clinical presentation is very typical, you know there is a single gene involved or a triple repeat, you can send for that and you will get a diagnosis. Sometimes there may be multiple genes involved, so you first test for the common, but if it comes negative, then you may have to send for the NGS panel or do a chromosomal microarray or something like that. And sometimes there is non-specific concerns, like there is just developmental delay in epilepsy. Now that can have hundreds of diagnoses. You have a short stature, it could be endocrinal, it could be skeletal dysplasia. So in such cases, you have to decide whether first you need to do a microarray or you have to go ahead and do a trio, exome or a genome. Genome, I would still say that we can wait, but trio whole exome definitely has got a much better pickup rate than doing a single exon. Okay, and then if even that is negative, so in spite of doing a microarray, in spite of doing a trio exome, there are still chances that it can be a non-report, and that is the time probably we have to go for an academic or a research efforts to get a diagnosis if the family is keen and if the clinician is keen on that side. Okay, so yes, mutation analysis 100% genotype phenotype correlation. It will reveal the primary cause. It has a diagnostic and prognostic value. So once you are final, you are having a diagnosis, you can explain to them how the child will go forward. So if you have found a microdeletion syndrome, you can go to the websites, you can go to the support groups, and they can help in prognostication. And personalized medicine, again, I have told that it is important because the drugs are based on this precise mutation. And obviously, we know the importance of prenatal diagnostics. So implication is for the individual as well as the family itself. So diagnosis, I cannot emphasize more than this, that yes, genetic diagnosis is very important. But sometimes it's better not to diagnose at all. You know, a previous child with suspected Down syndrome, so you are just keeping it as suspected, but based on that suspicion, the prenatal will be done in the next baby, that okay, you test for Down syndrome. So sometimes you can just write the features that this was present. We don't know the diagnosis. So it is better to have no diagnosis than having a wrong diagnosis, which can implicate or change the reproductive decisions. So this is probably a term which many people don't like, but I feel that education is very important. Just like what we are having just now, it becomes very important that we have to keep upgrading our knowledge. We have to know what to order, and once we receive the result, how to interpret it, because it may be misunderstood or misused and wrong information should go. Contents of the report, I have again informed that how it can if we are not familiar with it, we can give wrong information to the patient. Clinical validity, whether they are matching the phenotype, and clinical utility, whether the result is likely to benefit the patient. If even after testing for autism, you are spending a lakh of rupees and diagnosis is still autism, and you are not going to change any management, some patient may not agree for the testing, right? So we have to understand that patient's concept of why he wants to do the genetic testing. If they are not ready to do it, then also we should be okay with it. So, and most importantly, which madam also had told, that we have to be aware of the ethical considerations. So pre-test, post-test counseling, as ma'am had told in the starting itself, that it is really very important to take an informed consent. because patients are getting aware. They know their legal rights, and they know that how genetics can change everything for them and their family. Okay, they can have familial problems if they know that it is X-linked and only male members are going to get affected. Then they may lead to divorce, and then sometimes job implications are there, insurance are there. So every test, genetic test, even if storing a DNA, taking a photograph, is all after having a written informed consent. So whatever you're counseling, you have a record of it and get the signatures. You have to create patient awareness that how, whether it is going to be benefit. So doing, a mother has been found to have a mutation in BRCA1, she has breast cancer. Will you do it for a small 8-year-old minor girl? Right? So there are many ethical issues, there are many various viewpoints in this, but unfortunately, no specific guidelines exist. So country to country, state to state, institution to everybody has their own policies and they may vary. So I think the societies like Bangladesh Genetics Society, they should come together and have certain guidelines for this. More importantly, it is that it will increase the anxiety because of this variance of uncertain significance, which we also are not aware. Then how will the patient be understanding that what is this? There is something, but still we are not getting a result. So how to deal with it? And then there are incidental findings. So you are doing screening of a newborn and you find a good variant or a very pathogenic variant in a cancer gene. Will you report that? Will you tell the parent at birth that your child is going to get an adult onset disorder? So all these incidental findings, various of uncertain significance, are big ethical consideration, and everybody should have their understanding and where they have to put their foot down, that no, we will not want these incidental findings, or you can always take a consent and get those incidental findings for your patient. So it is not just in genetics, but in many other ways, NGS is being applied, and it will come together like metagenomics or epigenetics. In every way, NGS is the test forward. So just summarizing that it is coming up in a big way, but we have to know that the main challenges is not in the result. It is in the interpretation of the variants, and the genomic and phenotypic data has to be, we have to have good databases to understand the responsibilities and what the response or could be coming out of these data. And as we are continuing this education, this should continue so that the newer generation and those who are interested should keep learning and implicating or involving them in their day-to-day clinical practice. So just the take-home message is that all patients should be tested. Sometimes monitoring may be a problem, but they can be given a timeline that, okay, don't do it immediately, but before you plan the next pregnancy or before taking any reproductive decision, maybe in a month, 2 months, or you can, slowly you can talk to the labs, you can take some discounts and just get them evaluated. And classification, prognosis, treatment, everything ultimately depends on the molecular diagnosis and to prevent recurrence, and obviously for alternative reproductive options. So whether they want to do a prenatal testing, or they want to go for IVF pregnancy, or they want to do a PGS, PGD, all this will also depend on the final clinical diagnosis. So I just want to welcome you to the era of next generation sequencing. I thank you for your patience to hear me for so long. I really hope that I have been able to do justice to the topic. We are finishing 4 webinars on basics of genetics and genetic testing, but I can request Dr. Saha if there is any burning topic which needs to be covered, as he had requested for NIPT. Similarly, if there are any more topics, then we can definitely go forward to it. I thank the organizers and my team for always supporting me and being there for me, and obviously my teacher and my guide, Dr. Shubha. Thank you all, and I hope we have time for some questions, or it's too late.
Thank you, Dr. Sheetal Sharda, for your excellent talk. It was a very long discussion, actually.
Yes, I just
Thank
saw
you
that.
for your
Sorry
effort,
for that.
and thank you for the audience also.
They are still there or everybody has left? uh, okay.
Sheetal, we have a couple of questions in the Q&A window. Maybe you can address them.
Okay, so wild type is normal. Can't changes also occur after transcription. QC failure. Okay, wild type is, I may say yes, normal, because we do it like wild type and mutant type. So wild type means according to the reference sequence, if you want to know. So wild type means as per the reference sequence, which is common in the population. So that is a wild type. Yes, changes can occur after transcription. So there are post-transcriptional, post-translational changes. That's why we say that NGS or doing the DNA is not 100%. Genomics is much more than just doing DNA analysis. There is a whole metabolome which has to be done. There is a transcriptome which has to be done. There are epigenetic mechanisms. So there are many changes which can occur even after transcription, which can cause. QC failure is quality check. So there are various levels where we have to check the quality of the DNA, whether it has processed well, whether PCR has processed well, whether library has been prepared well, whether the run is gone through, so whether the male and the female gender is matching as per the request form. So this is what a QC or a quality check failure is. Yes, I agree. Too long single lecture. Even I am feeling tired now. I'm sorry, but I go into the teaching mode and Dr. Saha knows about it. So next time I'll be sure to make it very small, whatever it is. Very elaborative. Can you cite any example of affecting fertility of the patient and this like disorders affecting fertility? Yes, there are many genes which we are also now making a panel for primary infertility, secondary infertility, male infertility, female infertility, and yes, they may be transmitted. So there are a huge list. If you can send me a mail, we can share that with you. DMD treatment, 2 out of 33 trials successful. I am really not sure about the latest statistics on that. This is because recently I had prepared the slides and these two was what I came across, and it is also being given. One of my patient literally went to America to just have a try at this. So, but I'm not sure about the other. They may be more than 34 trials, so we have to check. So all features must be noted and informed. Yes, pseudogene. Pseudogene is a gene which is very similar to the true gene, exact replica, but it will not have a protein. So it will not be coding a protein. It is just there. So exact sequence match, but it will not be translating into a protein. Genetic testing is available in our country. Exactly available, I am not sure, but yes, logistics is there to take samples from your country and get tested to anywhere in the world. We can always explore that sequencing or MLPA. There might be some labs, but I'm not aware of that. Probably Professor Saha can answer that question.
Actually, we don't have so much lab, but we have the facility to collect the sample and send it to somewhere else like you and other places.
Okay, sir. Please comment on the advancement in clinical gene therapy. My God, this is a big webinar. I think more than 2 sessions itself, because a lot is happening in clinical gene therapy. Up to what trials they have gone and what are the replications. So right from starting from for the primary immunodeficiency, ADA gene deficiency, till today for DMD. Just yesterday we had a journal club in which for Dravet syndrome they have done this tango technique. So the name itself is very tango, but many different techniques, so nonsense mediated decay or exon skipping. So in which gene or which disorder which can work, and now CRISPR is coming up in a big way. So yes, it is going a big way, but we have to be skeptical before it comes into clinical practice. The last question probably is importance of introns in diseases. Yes, thank you for bringing it up. We have been concentrating on coding regions for a long time. The introns are feeling bad about it, but unfortunately, NGS does pick up intronic variants, but we don't have literature to predict. The way we have softwares to predict exonic variants, we don't have predictions for intronic variants. People are working up, that's why they go for whole genome. But previous literature or previous reports where they used to do Sanger and they have found intronic variants, they have done functional studies. If those variants are there, they will be covered in the NGS. That you have to get a confirmation from the lab whether reported intronic variants are covered or not. Okay, so the newer pipelines which are coming up do have the reported intronic variants also, and yes, they also can cause disease phenotype. So they are not to be ignored. They are not junk DNA anymore. So they are important. Okay. I think I have covered all the questions right from the start. Yes.
I think so.
Thank you everyone for thanking me, but I'm sorry for taking so much of your evening. And
Yamin,
Yes, sir.
please shift your mic to Professor Shahidullah sir for making a concluding remarks.
Okay, sir. Now I request Professor Mohammad Shahidullah sir to make the concluding remarks and to conclude this today's webinar. Sir,
Thank you,
please.
Yamin, and thank you, Professor Naran. First of all, I'd like to thank Sheetal Sharda for, though exhaustive, but I think this is very, very informative, very clear, and a lot of information, particularly those who are interested in genetics. I think they are largely being benefited, and others also, they have known that where the genetics has gone today. This is not the end of the journey, and we need to listen again and again. And Bangladesh Society for Medical Genetics would be is indebted to you, and will be looking forward to you for your future cooperation, giving us the light of your knowledge to our audience. And with that, I'd like to thank you and also the my Secretary General Naran Shah, and also Yamin today, the anchor, and those who have been with us during the whole presentation. Thank you from my side.
thank you, sir, for the kind words. And I will definitely try to make it smaller from next time. I really lost track of my time.
No, I mean, it's fine, but if you can little bit make it shorter, then people will have ample chance to make a lot of questions.
Yes.
Because that's
but
more important.
When I'm not aware of the audience, I like to go from the basics. So if I know that they know a certain type, then I can shorten out or go faster on certain slides. But
today, actually, with this, the fourth one, some basics have been already been shared. Next time when we'll be inviting you, will be something like that, that you take an issue, discuss, and then, we have question and answer.
Sure. We can do that.
Okay.
Okay, so that's about it for our webinar. So thank you very much for the audience for for patiently listening into Dr. Sheetal. Thank you, Dr. Sheetal, for taking time off and giving us this very important topic. And on behalf of MedGenome, I'd like to thank Dr. Saha for helping organize this webinar, and also the dignitaries who took time off from their schedule to be with us. And also the special thanks to the audience for their patience. And if you have any questions or comments, you can write into us through our partners in Bangladesh, Genetic Solutions Bangladesh. The details are given over here. So if you have any test requirements also, you can contact them and they will get in touch with us and help out with the logistics and everything. And if you need more information, you can visit us on our website, medgenome.com, for more information on all our products on exome sequencing, chromosomal microarray, NIPT, etc. And so just a reminder that we be sending out a recording of this webinar in about a week or so. So look out for that in your inbox. So thank you again for your time and your patience. And we look forward to seeing you on the next webinar on November 6th. That will be on NIPT, non-invasive prenatal testing. So until then, have a nice day and enjoy your weekend. Thank you.
Thank you. Good
Thank
night.
you. Thank
Thank you.
you.
Thank you. Good night.
Thank you, Joseph. Thank you very much.
Thank you.